Piccolo is a European project funded by the Horizon 2020 programme and supported by the European technology platform Photonics 21. This project proposes the development of a photonic endoscope to improve the in vivo diagnosis of colorectal cancer and to support clinical decision. In order to develop a project of these characteristics, 9 partners from 5 different countries have been involved. These are 3 partners from Spain, el Centro de Cirugía de Mínima Invasión Jesús Usón, Tecnalia and BioF. 2 partners from the United Kingdom, Imperial College and M2 Lasers. 1 partner from Ireland, Tyndall. 2 partners from Italy, Light for Tech and Lens. And 1 partner from Germany, Karl Stolz. Colorectal cancer represents approximately 10% of all cancers, being the 3rd most common in men and the 2nd in women, causing almost 700,000 deaths per year. Although the average age for its diagnosis is 65, it also occurs in young people and affects men and women almost equally. With this high incidence and mortality rate, it is essential to perform an early detection of the disease. It has been shown that a cure rate of 90% can be achieved if the disease is diagnosed in the early stages. To achieve the disease's early detection, health providers develop early diagnosis programs, being colonoscopy the standard method to detect and remove all the polyps, despite the fact that almost half of them have no malignant potential. This means a great effort, both economically and in time, for the healthcare system and a risk of injury to the patient. In addition, after the polyp removal, there are cases in which residues remain, and there are even certain recurrences rates due to its incorrect elimination. It is not only interesting to know what is seen of the polyp, but also what is unseen. It is necessary to determine the depth of invasion to achieve a better diagnosis. Almost 30% of these polyps, in particular flat ones, are not detected. This is because they are hidden in the colon's folds or are not well distinguished from its healthy tissue. Once all these needs have been identified, the aim of the Piccolo project is the development of photonic-based endoscope technologies. Providing an in vivo optical biopsy will improve the diagnosis of colorectal cancer that will allow to obtain the diagnosis in real time. This will reduce both cost and time, as well as leaving the benign polyps in the colon. The operation of the device will be the following. With white light, the polyps will be detected with the support of a computer program based on deep learning algorithms, which intends to reduce the missing polyp rate. The aim is to obtain both structural and functional information from the polyp tissue through optical coherence tomography and multi-photon tomography photonic technologies. This will allow a computer program based on deep learning algorithms to classify whether the polyp is benign or malign. In addition, the depth of the polyp could be established, as well as to evaluate its safe removal. The role of the Centro de Cirugía de Mínima Invasión in the project is the contribution to the detection of medical and clinical needs, both for the procedure and the technology currently used in colonoscopies. From a technical point of view, the Centro de Cirugía de Mínima Invasión will be responsible for the development of the software that will help with the automatic detection of polyps while scanning the colon with white light. From a clinical point of view, the Centro de Cirugía de Mínima Invasión will carry out the device's validation test on animal model, specifically rats with both benign and cancerous polyps. In addition, el Centro de Cirugía de Mínima Invasión will perform the necessary laboratory tests on rabbit and pigs models to ensure the safety of the device. The results of the Piccolo project will mean a significant improvement in the European diagnostic sector and therefore in related companies, which in turn will significantly improve the EU's healthcare systems. This endoscopic device will not replace any clinical staff. However, this will serve as support and assistance for the detection and decision-making in the diagnosis of colorectal cancer. The final clinical users will benefit from a system that improves clinical decisions through immediate additional information which is not available on current clinical procedures and imaging systems. From the patient's point of view, they will benefit from an improvement in their diagnosis, the non-elimination of benign polyps, a decrease in the number of re-interventions, and the time to obtain the diagnosis, which goes from days to minutes. This will allow, on the one hand, to limit the psychological stress of such waiting, and on the other hand, to improve patients' prognosis and quality of life. This will reduce the time in hospital and traumas. The healthcare system will also benefit from cost savings. This is because with the new device, histological analysis of benign polyps biopsies will not be performed. In addition, by improving the prognosis and patients' treatments, the cost of intervention and re-interventions, as well as patients' hospital stay, will be reduced. Finally, the companies and technological institutions involved in the project will have a new technological asset to grow and achieve a better position in the biophotonics market. Moreover, the developed system will allow other companies to develop new biomedical photonic-based devices as well as new software for their control.